The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon, everyone. My name is Jeffrey Dewey, and I am an Extension Educator with UMass Extension. Thank you for joining us today for TickTalk with TickReport webinar. This webinar is co-sponsored by UMass Extension Landscape, Nursery and Open Forestry Program and UMass Laboratory of Medical Zoology. The webinar will be recorded and archived on the UMass Extension website. Our speaker for this webinar is Dr. Stephen Rich, who is Professor of Microbiology and Director of University of Massachusetts Laboratory of Medical Zoology. In today's webinar, Dr. Rich will provide an update from the Tick report at UMass Amherst. If you have any question during this webinar, please type it in the question box on your control panel, and Dr. Rich will answer the questions at the end of the webinar. After the webinar, there will be a short survey. Please complete the survey before exiting the webinar. And now, without any further ado, here is Dr. Rich.
Thank you, Jeffrey. Do I need to click change presenter, or does that happen on your end?
You need to. Okay.
Great. Everyone seeing my first slide?
I can see, yes.
Okay, great. Thanks so much. Thanks, Jeffrey. And thank you, everyone, for coming this afternoon. I know these are extraordinary conditions, and I appreciate that so many of you tuned in and going to listen to what we have to talk about today. What I really want to do is give you a little update on where we stand at the University of Massachusetts with TickReport, in particular the tick testing laboratory, and give you a little idea of where we stand in terms of tick season. But I want to leave plenty of room to receive questions and respond to questions. So one of the pieces of feedback we've had is that we haven't had these terribly interactive, these TickTalks be terribly interactive in that we've answered questions in real time. But I want to try to do some of that today because I know you all have questions, and I hope I can have some answers to some of them. So I'll pause throughout and get your questions that way. So the first news I want to give you about TickReport, the tick testing service at UMass is fully operational. So much of the university has been drawn down. All teaching is now being done remotely. I teach my classes online via Zoom, which we're all learning. And hopefully by the end of the semester, I'll be good at it. And we are continuing to run the lab under a special contingency plan, a continuity plan that's allowing us to continue to receive and test ticks. But we do have a slightly different system. As I've indicated here, we have a special infection control plan in place that keeps our staff isolated one from the other just so that they have social contact to keep them safe. And they're working in separate space and staggered shifts. I want to just say a very positive word about those members of the staff. Those are all full-time employees, full-time staff of the University of Massachusetts working in my lab, and each of them has the opportunity to work remotely from home on other tasks, but each of them has felt that their job in TickReport and testing your ticks is vitally important, and so they've elected to come in and to travel from their homes to the laboratory to do the work, and I'm very appreciative of that, and I'm sure we all are. As far as what our service is providing, we're doing everything we've always done in terms of TickReport. We're still guaranteeing a 1 to 3 day turnaround. As volumes go up and without the ability to hire on new spring staff, we're not sure how long we'll be able to maintain that, but we're doing everything that we possibly can to get the ticks turned around, the results turned around as quickly as possible. The other thing I want to mention is that we do not receive at this point, for those of you that live locally to the lab, close to the 01002 zip code, some of you have grown accustomed to bringing your ticks in and dropping them off in the lab. You can see a smiling face there and have someone to communicate with. That office is closed. In fact, the building is locked and much of the campus is under a lockdown because most staff in most laboratories and offices aren't present. So there really is no opportunity for you to drop your tick off. So we have to rely on couriers. As usual, we recommend FedEx or UPS. U.S. Postal Service works as well, although we have had some problem with receiving those specimens through U.S. Postal Service. So FedEx and USPS or UPS are the best ways to get things. I also want to tell you that our communication program, that is our engaged outreach programs, are fully functional. So most of the people that are working on that program are working remotely from their homes, but we're still able to interact by real-time chats on the webpage, TickReport.com. Again, that's a place that you can go and ask your questions, any time of day, and get answers to your questions about ticks and/or TickReport and/or tick testing, or just generally about tick-borne diseases. The way the real-time chat works is we staff it during the business hours, basically 9 a.m. to 9 p.m. or so. If you leave a message there off hours in the middle of the night, it will go to email, and we'll check it the first thing the next morning. So either way, we're getting pretty quick turnaround, and I'll show you some of the stats from that later on. We also have email. You can email us your questions and we'll respond to them. I'd ask that you not try to search my email and ask me, because we have staff specifically ready to answer these kinds of questions. I'm happy to do it too. It's just my email box is generally flooded with other things, including the 327 students in my class. And so it may be harder for me to get back to you if you leave an email. Likewise, if you call the office number or my office number, the TickReport office number or my office number, chances are there'll be a delay in response because we're not going into those offices. And then if it goes to voicemail, we'll check it as we can, but it's not a very reliable way to reach us. The last thing I had there on the list of contact for us is the Zoom. So we've taken on Zoom as a university. That's how we're delivering all our instruction now. And we're happy to figure out how we might be able to do Zoom to provide some of the outreach talks that we usually do in person. Paul Killinger is listening in today, and he or I could take your emails through info@TickReport if you're interested in having us speak to your group. We often go out and do these talks in person. Since we're in a condition where we can't do that, we're happy to figure out if there's a way to do such as this webinar to your group. So those are the updates on TickReport. And as I said before, please feel free to send us questions. I can monitor and see if there's any questions coming through. Nothing at this time. So let me go on and let me talk about the 300-pound gorilla in the room. I don't want to spend time talking about COVID-19, but and probably many of us are glued to television news or internet sources for information. I did just want to show a couple of slides that are up to date as of today from the CDC. These are statistics for COVID. And really the one I want to draw your attention to is, I don't have a cursor here. Let me see. I'm not able to locate my cursor on here, but the column I'm looking at is the column in the center that says the percent of expected deaths. This caught my eye when I was perusing these statistics. What those numbers represent is that is the percent of deaths that are being experienced in this week compared to the 2017 to 2019 total for deaths. So if you look down that column, you see that the number of deaths, the percentage of deaths this year, from all means, from all causes, has actually gone down. So it's an interesting phenomenon in that probably what's built in there is the fact that people aren't driving their cars, so we aren't having as many car accidents. People aren't having as many incidents out of the home, accidental deaths, which are a major cause of death. And so the death rate has actually gone down, ironically, during this period. And it holds true also when you look across the age structures. I don't want to spend too much time about that because I don't really know what's underlying there, and it's really a question for epidemiologists. But I draw it to our attention because it's a good point to remember that while we're experiencing this extreme pandemic situation with COVID-19 or SARS-CoV-2, we have to remember that all the other health risks that we typically would have in April are still present, including ticks and tick bites. And if anything, we are seeing an increase in the number of ticks submitted to our laboratory, we think in large part because as people are now finding themselves in a situation where they're confined to home, they're not going into work and or school, and just trying to maintain good health and good sanity, people are going outdoors perhaps more than they typically do. And so we're seeing a lot more tick submissions, which we think reflects a lot more tick encounters and potentially a lot more tick-borne disease risk. So something to be aware of, that there is this great concern about the pandemic, but other concerns exist and persist in the context of that pandemic. So what I want to really focus on today is, we've talked about tick biology and we talked about timing and some of the life cycle stuff. I want to really dig down and just give you some of the data that we're seeing from TickReport. This is our online representation where you can, or online presentation where you can interface with us, get information about ticks and tick-borne diseases, but also submit a tick for testing if you should be bitten by a tick. And a reminder of what it is that we're doing here. We're trying to measure the exposure, which we think is the fundamental determinant of risk. So we know that there's different ways to measure risk and hazard, et cetera, that there's the hazard that we can look at based on the number of ticks that are out in the wild and where they're present and how infected they are, that we can measure risk as a realized number of individuals that become ill with things like Lyme disease, represented by the images in the picture on the right there. But really, what often gets overlooked, because it's not always easy to measure, is that exposure factor, the factors that contribute to those hazards leading to a realized risk. And so exposure in the context of tick-borne diseases is threefold. There are the tick diseases, tick species themselves. It's important always to remember that not all species of tick transmit all the different tick-borne diseases. In fact, most of them are quite species specific. And also that the duration of the feeding is of fundamental importance, because pathogen transmission does not happen instantaneously. It takes place over the course of the tick's feeding. And then, of course, there's the infection status of the individual tick that has bitten a person, because not all ticks are infected. For things like Babesia, for things like Borrelia burgdorferi, the causative agent of Lyme disease, about 50% of the ticks that we find here in the Northeast that are biting people, about 50% are infected. And if in a few more weeks, when the nymphs are active, that number will drop down to about 30%. But the risk is still considerable because the ticks tend to feed longer because they're smaller. And so putting together these three factors is the best way to determine what the hazard risks are. So I'm going to turn for a second here because there are a couple of questions. What's the best way to package a tick for testing? So that question's coming through. The best thing we recommend is, when you remove the tick from your dog, your pet, your child, yourself, place it in a small Ziploc bag if you have one on hand, seal the bag, and then place that sealed bag inside an envelope. Doesn't need to be anything fancy. If you have a padded envelope nearby, that's okay. If not, you can send it without. And then just drop it in the mail, or drop it to with the courier, UPS or FedEx. Or if you're going to use USPS, first class mail is okay, although we've experienced some problems. Probably better to use priority, or best yet to use express mail. So I want to move on to the just to remind you why it's so important to understand that second risk target exposure factor that we talked about, which is the duration of feeding. I remind you that when ticks feed, they have a very lengthy feeding process. they attach, and then there's an exchange of fluids between the tick and the person on, or the individual on which it's feeding. And the flow of those fluids between the tick and the individual is unidirectional on both sides. So on one side, you have blood that flows from the individual being bitten into the tick and goes into the midgut. And then the stuff that's coming out of the tick is coming from the salivary glands into the site of that feeding lesion. And when we consider the path that pathogens take in those two one-ways, we find that the pathogen actually comes into the midgut with the feeding blood and remains there for a great deal of time while the blood is being digested. So a tick that might have fed last summer would keep its pathogen in the midgut through the succeeding seasons, and then only in the following season, when it started to feed again, would it make the migration from the midgut over to the salivary glands, where it's then ready to be launched into the next individual it feeds on. So it's fundamentally important to understand that it's only after a degree of feeding has taken place, and there's still some room for understanding exactly what that window is, but the best indications are that it doesn't take place for things like Borrelia until sometime at least after 24 to 48 hours, and that for other pathogens it might be slightly shorter. Now, some people will push back and say, no, I know I got infected from a tick that fed much shorter period of time. Well, these are averages, and so there's a tail in those distributions, and they should not be taken as single points, but as representations of the mean. But nonetheless, feeding time is of fundamental importance for the transmission of pathogens out of a tick. And because we can look at the ticks, change in dimensions as it feeds, we can determine or approximate how long the tick is fed. Here's a feeding diagram showing unfed nymphs across the top, unfed adults across the bottom, and you can see that the risk of pathogen transmission, if it's an infected tick of the right species, increases with that feeding time. So another question is coming through. Can you test partial or broken ticks, or do you need it to be filled with blood? Taking those questions separately, we can test partial ticks. So it's not uncommon when people remove a tick that they'll lose part of the mouth parts. The most distal end of the tip of the mouth parts might get broken off as they're removing it. We can still test the remainder of the tick that you have in the forceps. Send that in, we'll test it. And in fact, that's the portion that probably contains a bigger chunk of the salivary glands and or chunks of the pieces of the midgut, which is where we really are looking for the pathogens. Do the ticks need to be filled with blood? So there is some of our counterpart laboratories, for example, in Connecticut, they will only test ticks that are engorged or ticks that are fully fed. We'll test ticks that are flat or engorged because we understand that people want to understand the risk. We recognize that the risk of an engorged tick, if it's infected, are higher than the risk of a flat tick or an unfed tick, even if that tick is infected. Nonetheless, we'll test any tick that's sent to us to give you that information about that component of risk. The question that can ticks transmit Lyme in the fall and all other seasons? There's an interesting thing that we've encountered recently, that people are under the impression that adult deer ticks do not transmit pathogens. That's not true. Adult ticks transmit Lyme disease and other pathogens. In some cases, they have a higher infection rate, and if they're allowed to feed for the same duration, they actually have a higher probability of transmitting infection. Usually, they don't feed long enough because the ticks are larger and they're detected before they have the ability to feed to that point where pathogen transmission takes place. Nonetheless, nymphal ticks and adult ticks can both transmit. They can transmit in the spring, they can transmit in the summer, they can transmit in the fall, they can transmit in the winter if they're still active. And I'm going to show you in a moment the more active periods for those different life stages. But yes, ticks can transmit diseases in the fall, they can transmit the same diseases in the spring. So those risk assessments that we're making, those exposure assessments we're making, give you pretty good information about your exposure risk, whether the tick is positive or negative, whether it's fed sufficiently or not, and whether it's the right species or not. That's all part of a tick report. And we want people to understand that it's the whole tick report and not just the positive, negative for the pathogens that provide the best indications of exposure. And these are valuable risk assessments for individuals. And when taken in aggregate, they constitute a very nice passive surveillance that allows us to tell things like when ticks are biting people, who's getting bit, et cetera, et cetera. And I've shown you some of those surveys, some of those results of that surveillance in the past. I want to just use that as a springboard to talk a little bit about what's happening right now, particularly here in the Northeast, since I'm assuming that most of the 200 or so people that are listening to me at present are from the Northeast, and so this is appropriate for those folks. So I'll talk about the importance of exposure risk in time and space as we understand it from this passive surveillance. So just a bit first about the geography. The black-legged ticks, which are the ones that are of highest interest to us in terms of the transmission of, they transmit Lyme disease, the most common pathogen here in the Northeast. And there's the distribution, mostly east of the Mississippi, some Oklahoma and Texas as well, but a Northeast distribution with much of the transmission of Lyme disease happening in the Northeastern United States and the upper Midwest. Lone Star ticks are ticks that have been generally thought of being further in the Southeast, but as I'll show you in a moment, we're really seeing an expanding range, and we're finding ticks further and further northward each year. And so that's clearly a tick that's expanding its range. The American dog tick has probably been around the longest, or been one of known colloquially for the longest. And it has a distribution, again, throughout much of the eastern United States. There are also pockets on the West Coast, which may in fact be a separate subspecies. It's somewhat debatable, but nonetheless, the American dog tick is distributed as such. And that comprises the three major groups of major species of human-biting ticks in North America. Not the only ones, but the most predominant ones. And each of them is associated with different sets of pathogens. So this is another question that comes up occasionally. While only deer ticks transmit Lyme borreliosis, the agents of Lyme disease, they also transmit Borrelia miyamotoi, the agent of babesiosis, Anaplasma phagocytophilum, which is the agent of anaplasmosis, and also a deer tick virus or Powassan virus type 2. So many pathogens from a single species of tick. The dog tick and the Lone Star tick pose no risk of Lyme disease or very little of Babesia or Anaplasma, but they do have their own associated pathogens, but by and large to a lesser extent. So dog ticks are known to be associated with tularemia. We had a tularemia positive tick sent to us from California about 3 weeks ago now. Rocky-Mountain spotted fever can be associated with dog ticks, also the brown dog tick, which is a less common human-biting species. And then Lone Star ticks have their own set of associated pathogens, including ehrlichiosis, which is related to anaplasmosis, but a different bacterium. Things like SARI, the southern tick-associated rash illness, and of course the alpha-gal allergy, the red meat allergy, which we're learning more and more about as time goes on. It's clearly going to be important to understand the distribution of Lone Star tick bites and the degree to which those ticks are driving the etiology of tick-borne red meat allergies. We're working with some of the immunologists that have discovered that, made those initial discoveries, and trying to determine what the entomological risk factors are for that. And we'll probably be reaching out to those of you in TickReport that have been bitten by Lone Star ticks in the past to ask you if you're willing to participate in some follow-up studies in that regard, epidemiological studies. Turning my attention here now for a couple of questions. Does the pathogens migration from midgut to salivary glands happen over a few days during feeding or over a whole season? So the movement from the midgut to the salivary glands, they remain in the midgut after the tick is fed. And the tick will then digest that blood meal. No migration of pathogens occurs from the midgut and to the salivary glands while they're digesting the past blood meal, which can take months and months. And the migration from the midgut to the salivary glands only happens on the next feeding. And in fact, it's some of the components of that next blood meal that the pathogen is going to use to make its way across the boundary from the midgut through the hemocoel, which is the open circulatory system of the tick, and then back into the salivary glands. So it's a very good question. The migration of pathogens from midgut to salivary glands happens only upon the next feeding and not during the season when it's digesting that blood meal. Thank you for that question. Question is also coming, is it true that dog ticks are less likely to infect or carry pathogens? It is true that dog ticks are less likely to be infected and so therefore less likely to transmit their pathogens. That's not a 0% chance of infection, but it is a relatively low compared to, for example, Lyme disease. So tularemia we find to be much less than 2%, usually less than 1% nationally, whereas Lyme borreliosis we find to be not uncommonly greater than 50%. So yes, dog ticks have a lower association or lower percentage of tick-borne infections. Okay, returning to the presentation. So here's a pattern, and this is data for, sorry, just moving the screen here. These are data for a couple of years ago. I'm going to show you a series of the 2017, the 2018, and 2019 data. And what I want to show here is just the pattern, and then I'll show you them all in a single figure. So this is the seasonal activity for deer ticks as we've received them here in TickReport. So you notice there's a spring peak, the green bars of adults, and then a secondary fall peak of adults starting in late October. There's also a nymphal peak that you can see here that corresponds to about another month from now, starting in late May, reaches a peak in June and then drops off through the summer. That's the nymphal peak, and then a very low peak of larvae in the background. And that's probably just because those ticks tend to be so small. They probably tend to very often to feed in the leaf litter on mice, etc. And when they're feeding on people or our pets, they often go unnoticed because they're so very small. So you can see that pattern, the bimodal distribution of the adult ticks in the spring and fall. And so if we were to look right now at where we are, and I'm again speaking most to those of us in the Northeast, if we are right here in mid-ish spring, let's say, or early to mid-spring, what we are seeing in terms of deer tick activity is the adult activity period, and we will in a couple of weeks start to see the activity period of the nymphs. So right now it's adult ticks. The big, larger, the males are all dark colored, whereas the females have a orangish red margin at the back of their bodies. Those are the ones that are most typically feeding us right now. And this is just to tie that in with the life cycle stories we've told before. See, these adult ticks that we're seeing right now are actually from the 2018 cohort of ticks that were hatched back in spring, summer of 2018, larvae, nymphal, adult over a 2-year life cycle. And the nymphs that we'll see in a couple of weeks are actually the 2019 nymphs, which were hatched last spring, summer. And they will come out, as we said before, starting in mid-May and then extending through June, and then later they'll become the adults for the next generation. And the 2020 cohort, which is going to start this summer, will be the eggs that get laid later on as we get to the end of the spring and early summer. Those are the adult ticks that are feeding now, mostly on deer in the wild, and will fall off and lay their eggs to start the 3 cycles, the 3rd cycle. So there you see the 3 different cohorts, just to give you some idea of what we're experiencing right now, or right here, where we're looking at adults that started their lifetime 2 years ago. We're soon going to start to encounter nymphs that will overlap with those adults, and those are the nymphs that were hatched last year. And another thing to drive home here is that the reason we have Lyme disease is, you note that in a chronological, so here's the Julian calendar down here, we have the nymphs preceding the larvae each year, the nymphs preceding the larvae. And that's important because the nymphs are infected, they'll infect mice, and those mice will in turn infect the larvae, which are hatched initially without any infection from their egg stage. Pause here to see if there's any questions about that. Does the midgut to salivary gland migration have to happen for all pathogens and all tick species? So it has to happen for all the bacterial and protozoan pathogens, which is the lion's share of them. So for Borrelia and Anaplasma and Ehrlichia and tularemia, that transmission from midgut to salivary glands has to take place. There is a possibility that for some of the viral pathogens, that the viral particles remain on the mouth parts or in the salivary glands or in the hypostome, and so don't have to undergo that change, which might be one of the reasons that it's been shown in the literature that transmission of viral pathogens doesn't take hours and hours or even days, but can happen in as few as 15 minutes. So for some of the viral pathogens, there might be a different route of how those viruses make their way around, and I think there's more work to be done in that regard. How many times does a tick feed per year or season? Well, the best way to think about that is every tick feeds exactly once per life stage. So a larva eats once, a nymph eats once, and an adult eats once. So looking at the figure you have that I have on the screen presently, for the ticks that are living this 2018 cohort, they will feed once, twice, and a 3rd time as adults. Each of those stages feeds only once. So roughly 1 not per season, but per activity period, because some activity periods traverse several seasons. How many times does a tick feed per, can ticks transmit Bartonella? So I'm going to go out on a limb here, and I'm going to say that we've tested over 50,000 ticks in our laboratory for Bartonella and have detected no Bartonella infection in the ticks that we've tested. Now, there's a couple of caveats there, and I know there are other laboratories that are testing ticks and claiming they're finding Bartonella. There's a couple of different reasons that we could explain that. Some are, let me just, so a couple of different reasons we could explain that. One is the Bartonella test that we perform is very specifically for Bartonella henselae, the agent of cat scratch fever. We don't use an assay that will detect the genus Bartonella writ large or other related versions of that bacterium that may or may not be pathogens. We only focus on Bartonella henselae, which is the species that people are concerned about as having a tick-borne etiology. It may be that other laboratories use accidentally or perhaps by design, it's hard to say because they don't really reveal their methods. They may have assays that are more broadly construed and will detect some of those other species, but then they're not detecting Bartonella henselae, they're detecting Bartonella, which may or may not be a pathogen. They may be detecting bacteria that are soil bacteria that are on the surface of the tick and have no association with the tick bite itself. So our data suggests that there's very little evidence for a tick-borne etiology of Bartonella. So if people are experiencing illness from Bartonella, understand that that does not deny the claim that it doesn't have a tick-borne etiology does not deny that you may have been infected by another route. It also does not preclude the possibility that yours was a rare instance of a tick-borne etiology. I'm just telling you that based on what we've seen, there's very scarce data supporting the fact that Bartonella is transmitted by ticks in the northeastern United States. Question is, do male ticks also feed and transmit pathogens? The short answer is yes, they do feed, they can transmit pathogens. They don't generally do so, they don't generally do either, and that's because adult male ticks will take a small blood meal, but basically they spend their time looking for females where they will mate and then die. So they are not going to take that final full blood meal as an adult male. They may take a small one, and because they take a small blood meal, they have a very low probability. It means they feed for a short duration, a small possibility of transmitting pathogens. So while it's possible, it's rather unlikely that male ticks will bite and or transmit pathogens. How long can a tick live without feeding? Well, a good portion of a tick's life. So let me again point you back to the diagram that's still shown on the screen. Basically, you can think of the leftmost margin of each of these as representing the amount of time that a tick is actually feeding. And most of the bar here is actually time that a tick is not feeding. It's digesting its blood meal, but it's not feeding. So in the course of a 2-year lifespan, a tick probably spends maybe a couple days as a nymph, a couple days as a larvae, a few days as a nymph, and then maybe a week or longer as an adult. So maybe over the course of 2 years, it spends the better part of 2.5 weeks actually feeding. The rest of that time, it's digesting. It's not starving, but it can go a very, very long time without feeding, and very long relative to its own life cycle. How long can a tick live without feeding? Sorry. Is the 2020 season expected to be bad for ticks? Does a mild winter affect tick season? Well, I'd encourage you to go back and watch our last webinar. We talked about the role that weather plays in driving tick cycles, and it's not all about snow. So snow is one of the factors that provides insulation for ticks, but it's more about leaf litter, as you'll see back then. So we don't really try to make predictions about ticks based on the severity of the season. We know that, in fact, every year is going to be an important tick season, and often what people are experiencing as a rise or fall in the number of ticks is not a year-to-year variation, but actually seasonal variation. So let me see if the next slide going on will help us to answer that. So I think very often what people see is they're noticing in March of a particular year that maybe ticks are more abundant than they were in a previous year. All that is maybe a slightly earlier or a slightly later to the start to the tick season. And people often forget that there are these cycles that actually the tick season for deer ticks wanes off at the end of the summer. And so people are often led to say, oh, it's not a very serious tick year because this August I didn't see many ticks. When in point of fact, you see very few adult ticks in any August of any given year here in the Northeast. And so there's partially an illusory effect to thinking that there's changes from year to year. We think that ticks are pretty constant, and our TickReport data sort of shows that there might be some years where there's slightly more ticks or slightly less ticks, but not statistically meaningful. And so we don't say that this is going to be a bad tick year or a good tick year. It's just going to be a tick year, generally speaking. Let's see if there's another question we can answer in here. Is there a difference in transmission speed between nymphs and adults? There is a difference, and it has to do with the timing of the ticks feeding. So nymphal ticks generally complete their feeding in a matter of 3 or 4 days, whereas adult ticks take 7 to 10 days. And so the whole time frame for transmission is sort of slowed, if you will, in the adults relative to the nymphs. So continuing with this conversation, I'm going back to the slides now about the seasonal activity. If we look at 2019 data for the 3 species, deer ticks, dog ticks, and lone star ticks. If we look at the pattern over 2017, 18, and 19 for those 3 species, predominant human biting species, you see that there are some variations. Here the peak in the fall was a little bit more. 2017 peak was a little higher than we saw in 2018. Some of that may be reporting error, so the number of people that are sending us ticks or the number of people that are detecting ticks, or it could be in fact that there was a slightly higher number of questing ticks in this year. But important to notice that the numbers don't change substantially. So you're talking about a difference of a peak of 280 here versus, you know, something close to 700 in this one aberrant wink. So it only takes 1 tick to transmit pathogens, and so a difference of 300 ticks versus 700 ticks is probably not meaningful in terms of the way you'd want to go about assessing risk. Far more important to know that in these weeks of November, here, and here, the risk is much higher than a corresponding week in that same year in August or September in any given year. I think that's a much better way to look at it, not to look at year to year, whether 2017 is worse than 2018, but rather to look within an individual year and to know that the risk in January, while not 0, is much lower than the risk in April. Likewise, the risk in August, September is much lower than October, November. So I think that's a better way for us to be thinking about risk and its assessment. A related question has just come through. Are we seeing an increase in the rate of infected ticks from year to year? We're not seeing an increase per se where ticks are. What we're seeing, let me say that again. Where the ticks are present, we're not seeing an increase in the relative infection rate in those ticks. What we are seeing is that ticks are broadening their ranges. So I've said this a few times in places like upstate New York, the Adirondacks, northern Vermont, New Hampshire, upper northern Maine, we're seeing deer ticks expanding ranges, and to a certain extent lone star ticks expanding ranges to places they weren't present previously, and they're bringing with them the infections that they had in the more southerly and more coastal sites. So no, where ticks are established, they're not increasing their infection rates, but ticks are becoming newly established in new territories and presenting new risks in that regard. Here's just, again, data from 2017, 2019. These are TickReport data showing the distribution of lone star ticks. Now, these are points for people that have reported being bitten by a tick, and these are their home addresses. It's important to emphasize that we distinguish between the ticks, the site of the tick bite victim's home and the site where they actually encountered the tick bite. So in all likelihood, some of these bites, for example, out here in the West, are probably ticks that were associated with tick bites that were acquired someplace in the endemic range of the lone star ticks. And in a future talk, when we come back to talk about red meat allergy, which I'd like to line up to get a colleague to come in and talk with us about that, maybe in May or possibly June, we could talk about this distribution of lone star ticks. But clearly here in the Northeast, it's expanding its range. So when we look at the sites of origin of the tick, where the tick bite's actually occurring, we see that it's occurring, people are being bitten by lone star ticks further and further north than we would have seen 5 or 10 years ago for sure. So I really want to spend a little bit more time answering questions. So if you have questions, please send them across. These last couple of slides, I just want to show you the images. So when we get asked questions like this one that just came up about the increase in infection rates, what I would encourage people to do is to go to TickReport.com, find our stats page, which you can type in directly. The URL is TickReport.com/stats, STATS. You can also find a link to it from the front page, the landing page at TickReport.com. And you can enter your zip code. For example, I've picked out the town of Centerville, which is just a place loved by my family. And I've shown here the data for Centerville, Massachusetts for 2019, and the data for the state of Massachusetts in 2019. And so you can see how Centerville is a representation of the bigger population of Massachusetts. So in this case, I'm showing the multiplicity of infection, the number of ticks that were infected with 0, 1, 2, or even 3 pathogens, as shown by that very thin slice of pie. But you can do this on TickReport.com/stats. You can see the stats for individual pathogens, the presence, absence of Borrelia, Lyme disease, Anaplasma, Babesia in your zip code, in your state, or you can even select a series of zip codes. For example, if you wanted to enter the zip codes for your county, you could find this at a county level. We're working on improving our GIS tools so that you can also just enter the county yourselves. But it's not only possible to restrict it in terms of geography, you can also restrict it by time zone. So you could look within the past month, you could look over the past year, you could look over the past several years with our database going back to 2006. I will say that our data really only got dense enough starting probably around 2013 to provide any really meaningful epidemiological information for many areas, and certainly very dense here in the state of Massachusetts. Just another figure, another version of what you might find if you were to go and look at the individual infection rates for the town of Centerville and the state of Massachusetts on our stats page, where you see the total number of ticks submitted and the number of those ticks that are positive for a given pathogen. Anaplasma, for example, 258 ticks out of 3,770, 258 were positive. And the relative infection rate for things like Borrelia. So here you see why Lyme disease is of greater concern in terms of just the numbers, 1.3K out of 3.77K. Much smaller incidence of Borrelia miyamotoi. This thing, Borrelia mayoni, that's a subtype of Borrelia burgdorferi sensu lato that was identified in outside the Mayo Clinic, and we haven't found it outside of that state since its first discovery a couple of years ago. But nonetheless, we still test ticks for it to see if it's expanding its range. So you can go and look at these data, pull them down, and try that yourself. I'm going to see if I can switch over and actually show that what that would look like for you. So here is what you would come to if you were to look at our stats page. Paul, if you're on there, can you hear me and just give me a heads up that I'm showing now the stats page? Great, thank you. Okay, so I'll just take it back a step so that you can see how I got here. Here's the page that we're encouraging you to go to get resources and information about ticks and tick-borne diseases. Down below is the chat line where you could talk to Paul, as I just did, or Meredith, or other members of the team. If you go along the top, you find we have some FAQs. These answer a number of questions that we've encountered over the 14 or so years that we've been answering these questions. And then you can also find our statistic page by this link here. So I'm going to click the link, take you to the statistic page. And here's the viewable window where you can enter the boundaries of those dates that you'd like to look at. So I'm just going to leave it at the default, which is the past year, the past calendar year. And I'm going to change it to Massachusetts. And I'm going to enter the zip code for here in Amherst, because we do get a lot of ticks from Amherst, just because we've grown here first and foremost because of word of mouth. And I refresh the data. It crunches away for a second, and then you pulls up the graphs that show you the tick bite. Here's the age distribution of tick bites. Anywhere where we've seen a sufficient number of tick bites, human tick bites, we'll see this kind of age distribution. It turns out that college age and 20-somethings don't get bitten by ticks as often as those of us in the middle age group or those in the very young. So this age distribution is sort of uniform throughout the range. You can find the spots where people are finding the ticks, parts of the body. You can see the associated hosts. So most of the ticks that we are testing are coming from humans. That's just a bias of perhaps what people are most interested in having tested, but we do get a small percentage of ticks from cats and dogs. There's the percentage of tick species in the past year. Amblyoma is present, but obviously a smaller percentage relative to deer ticks. And I don't want to go through all this ad nauseum, but you can scroll through and you could find these statistics, and you could go back up and change it. For example, if I wanted to not have just a zip code, but I wanted to check, for example, everything that's going on in the state of Pennsylvania, I could do this. So we receive lots of ticks from Pennsylvania. There is another lab that tests in Pennsylvania. That's the lab that's finding the high number of Bartonella, high percentage of Bartonella. We test very few ticks. Very few people request Bartonella testing through our service, but as a research project, we went back and conducted a survey on, as I mentioned before, some 50,000 ticks and found none of them to be infected with Bartonella. So those are data for Pennsylvania, and you can do that for any state or combinations of states, and you can restrict it by species, etc. We're happy to make this resource available. We think it's important. It's a great way for us to give back to the community. It makes what you're doing also part of a citizen science project. So in addition to testing your own tick and getting information about your own risk, this data is being fed back into the general database. All of your private information is obviously kept private. It's never shared anywhere. But other people that might elect to not test a tick or decide not to test a tick for one reason or another can still get information about what's going on in their zip code or their home. Anything else I wanted to show here? Oh, and there's just, you can see the individual, you can see the types of ticks that we're receiving. There's the towns that are sending them. And you can scroll through these lists and look at things like the life stage and the number of tests that were applied, and etc., where you can find the outcomes and even see the images of the ticks that were sent to us, so you can get some idea of what ticks look like, because we like to emphasize that here's a fine example of a tick that doesn't look at all like the pretty pictures of the adult female Ixodes scapularis with that red margin. She's lost the pigmentation in the back part of her body as she's become replete with blood. The shape and size of her body's changed, and so this will give you some idea of what ticks look like after they've fed for an appreciable time. Okay, I'm going to switch back over to the PowerPoint and sort of finish up here. I just want to emphasize that a big part of what the TickReport laboratory does, a big part of what we're engaged in, is communicating to the public. We figure it's not just our mission to communicate to individuals that have tested their ticks. We also want to communicate with just the general public that has concerns about ticks and tick-borne diseases. And so we spend a lot of time trying to develop these different tools and outreach programs and library talks and talks to different nature groups. And as I said, we're willing to entertain the idea of figuring out how we can do Zoom meetings with your group. And this is just one small representation of what we've done in the past year in terms of, we have a program called Intercom, where we have these real-time chats and email chats with people that have asked questions, and it gives you some idea of the volume of calls and conversations that we have through the course of those several months. I'm going to glance back over here and see what questions we can have to answer in the last couple of minutes. Do dermacenter and amblyoma ticks have a similar 2-year life cycle? The answer is probably no. They seemingly have just a single life cycle, a single 1-year life cycle that doesn't overlap in the ways that you saw the cohort model. They seem to have a single peak in the charts as you were showing. Yeah, so what you're seeing is that single the larvae, nymph, and adults take the span of that life cycle occurring over a single year. Second questions, are ticks known to develop a tolerance for DEET or permethrin? So these are great questions. DEET is what we recommend as a tick repellent. So DEET applied to the skin will prevent ticks from feeding on that skin, although it requires frequent reapplication, probably every couple hours or every several hours. Permethrin is what we recommend for treating people's clothing. It's an acaricide, not a repellent, so it kills ticks. DEET does not kill ticks, it repels them. Permethrin kills ticks, it doesn't repel them. And so the question is, are there resistance to either of these things? We know no evidence of resistance to either one at present. It's not to say that it might not come about, but given the fact that ecologically we're probably, although it may not seem so to us, we're probably not big feeders of ticks. We're not important to tick survivorship, not as important as, for example, as mice and deer are on the wild. And since deer and mice aren't being sprayed with DEET or permethrin, the ticks aren't probably being exposed to the rate that it would drive and select for that kind of resistance. And as I say, at present, none has been reported or detected. Does the Lone Star tick, common name for amblyoma, indicate a geographic origin or other reason for the name? So the Lone Star has to do with the single spot in the back. I won't scroll back to those slides at this point, but it has a white pigmentation on the back of the female, and you can see that on any standard tick, or if you call TickReport.com, you can get one of our tick ID tools. We'll send you a photo tool. It's a clear laminate card with actual size colors and ticks and colors and sizes of ticks. We could have one of those cards sent to you. And the Lone Star tick gets its name from that lone star in the center of the back of the female. So I'm conscious of the time. It's now 1 p.m. I'm going to wrap up here. We can continue to receive your questions here on the webinar, which I guess is going to close momentarily. If you didn't get your question answered, or you still have a question you'd like to come up with, please, by all means, send it to info@TickReport.com or go to TickReport.com and enter the chat line and speak to someone in real time. They can answer those questions. You can also look at the FAQs if you're more inclined to go that way. The FAQs on our TickReport page. And we also are strong supporters of Tick Encounter. It's a good resource for information about ticks and tick-borne diseases. Go there. They're a partner site for us. They can also may have questions for you, although you won't get the real-time response that you can get at TickReport. So thank you very much. I realize that now in hindsight that I should have had the slide to tell you we're going to do this again in a month. It's going to be the corresponding Wednesday, the second Wednesday in the month of May. We'll come back. I haven't decided upon the topic. What I'd like to try to talk about is the alpha-gal red meat allergy, which I know is of growing concern to people. I'd like to get another expert to join me in that talk, but stay tuned. We'll definitely have another Tick Talk a month from now, and stay tuned for what that topic is. In the meantime, send your questions to TickReport. I thank you very much for your attendance today.